So welcome everybody to this first session of 4 for the AI2050 AI ML Tools for Drug Discovery webinar series. It's my great pleasure as part of H3D Foundation to host these sessions. And it's been an absolute honor to work with my colleagues at Cilia and H3D on crafting the material for these sessions over the last 2 training workshops. 3 actually. So we've held these workshops in West Africa, East Africa, and South Africa. And there's been such a high demand for them that we decided to open it up through a webinar series to try to share the core concepts of what we're doing with a broader audience. And so that's what these 4 sessions are all about. So I'm the program coordinator at H3D Foundation. And at H3D Foundation, we've been working closely with the Cilia open source to drive these sessions in Africa. And it's really close to the mission of what we do as collaborators. So normally I do quite a long introduction to really let you know who we are and how we work. But in the interest of time, I'm going to hand over straight away to my colleague, Jason Lojek at H3D, who's going to be leading this session. Feel free to pop any questions you have in the chat. We'll be monitoring it. As mentioned, everything will be shared after the session. So if there's anything that you're struggling with a little bit, we'll do our best to answer them either today after the session or in the subsequent sessions this week. Over to you, Jason. Thank you so much.
Great. Thanks, Nicola. Very good pronunciation of the surname. All right, here comes my slides. All right, so hello, everyone. My name is Dr. Jason Lojek. I am a researcher and investigator at the Holistic Drug Discovery and Development Center, or H3D, in Cape Town, South Africa. And yes, I'll be giving this first day of the workshop. So as we go through the days, just to note that please write down this link. We'll share it multiple times through the course of the days, but everything for the workshop, all of the links, all of the slides, the activity evaluation forms, and your quizzes afterwards will all be linked through this website, this link. So I'll show it for a few more seconds on the screen. But yes, also to echo Nicola, welcome back to everyone who has been a part of one of our in-house workshops before, but every time we hosted a workshop, we were completely inundated with the number of applications that were interested in doing the workshop. So here we're really trying to reach as many people as possible. And so this will be quite a compact and condensed course of all the key concepts and quite a lot of concepts to get through. But all of the material will be shared afterwards, so you can go through it again and make sure you understand each concept from each day. All right, so with that, let's get started. I'm going to today speak about some introduction, so a little bit about drug discovery and where the challenges are and why it's so difficult. Then I'll talk about a little bit about AI and sort of the general concepts about what it is. So we start demystifying this catch-all term. And then we'll start dealing with the main concept today, which is really about the data that we use to do our analysis and train our machine learning models and what the characteristics of that data should be. And then we'll look at plotting that data in chemical space and visualizing how compounds compare to each other and discuss some machine learning model aspects around that, and then finish with the overall machine learning model training pipeline, not for you to be able to train your own models, just to understand the concepts around what goes into training a model so that when you use them, you understand what to look for and what to watch out for. All right. So let's start with a little bit about drug discovery. Why do we need new medicines? Try and think of a reason or two. Hopefully some will come pretty easily. Some diseases that we face have no cures, so we need new medicines for them. Many diseases already have medicines, but maybe they're suboptimal. Maybe they're too expensive. For example, in TB, they require a 6-month treatment of combination treatment of 4 drugs. So if we can find new medicines to make that treatment better for the patient, more cost effective, then there's value that can be added there. We're constantly facing challenges around disease resistance. So our existing drugs, even if they were fantastic when first discovered and released, the pathogens that we deal with become resistant to those medicines, and so we need new drugs to fill the pipeline to keep that disease under control. And particularly in Africa, we know we face a lot of disease challenges, particularly with diseases that are neglected by the rest of the world, the global north. And so there's a real need for us to pursue this research on our continent to try and reduce that disease burden for the diseases that affect us. This is both across communicable diseases and non-communicable diseases. You'll see in this course, we'll tend to focus a bit more on the infectious disease aspect because that's where we tend to work the most. And then also just to note that these problems are only going to grow if nothing is done, because our population is expected to grow rapidly over the next few decades. And with climate change shifting disease patterns and where different pathogens are endemic or not, we also expect population changes on who's getting sick to vary. So now we'll start shifting a little bit into why is drug discovery hard? So the first thing I want you to think about is when we are talking about a medicine that we're going to give to an actual human, what kind of properties should such a medicine have? I'll give you 5 seconds just to think about what that could be. Right, so here's some of the properties. Obviously, we want it to be effective, so it needs to go in and kill the bug. If you're targeting a pathogen, it needs to treat that bug, kill it so that you make the patient healthy or inhibit a certain protein that's dysfunctional, etc. And that's only half the battle, where really a lot of drugs fail for various toxicity reasons. So you don't just want to kill everything, right? You want to be selective. You want to only target your pathogen of interest or your protein of interest and not hit other off targets, which might start to cause toxic events. Then there's other factors that go into this. So of course, cost is a factor. A lot of populations in Africa can't afford drastically expensive medicines. So ideally, you would like it to be in as few doses as possible, which is both tolerable for the patient and then cheaper to manufacture. Depending on the disease you're targeting, perhaps you want a particular route of administration, so orally through a tablet rather than an injection where maybe a hospital or a nurse is needed. Some medicines have a lot of stability issues, and perhaps we can't keep a cold chain in various communities. So ideally, you would like a medicine that is stable at a higher temperature and doesn't degrade. But overall, we can summarize these two categories into a medicine needs to be safe and it needs to be effective. So you need to treat the thing you want to treat, but it also needs to not cause harmful effects, or you need to at least minimize harmful effects as you go. And so how we do drug discovery, maybe you've seen a pipeline that looks like this, where looking at the industry as a whole, typically in the pre-discovery phase, you'll have tens of thousands, millions of compounds being screened for potential activity to work as a medicine. But only a few of those will be actually progressed into formal drug discovery projects. And so here in the drug discovery phase, this is, for example, where H3D spends all of its time. And across the industry, maybe you'll have a few thousand compounds where you're making chemical modifications to those compounds to try and optimize how potent it is, to optimize how soluble it is, how well it's cleared in the body, etc. And many, many compounds are going to fail because they do not have the right set of properties. Either they're not potent enough, or they're not soluble enough, or they're cleared too fast. But if you're very lucky, we would be very, very happy with a day where we progress a compound to the preclinical phase. And you can see this huge drop off in the number of compounds that make it through. And here you're looking at animal studies, looking for tox flags, making sure that the medicine is really safe before you actually put it into human clinical trials. And here the clinical trials are also in phases. Here you'll start with healthy patients in just a few, and here you're really focused on safety in phase 1. And then as you move further through, you're now starting to look for both safety and efficacy. And after all of this effort as an industry as a whole, when you started with thousands of compounds, even for every 10 compounds that make it into clinical trials, you only land up with one approved drug. So it really is a long, expensive process. And we're very worried about this concept called attrition, which is where you're losing compounds all through the pipeline because they're not going to make it as a medicine. This takes many years. So on average, anywhere between 10 to 15 years, costs anywhere between $1 to $3 billion to do this whole process. And so any improvements that we can make in this process is going to have value in terms of reducing the time and expense. One more slide around why drug discovery is difficult. I really like this question and blew my mind when I first heard it. So we don't know the answer, but what is the estimated number of molecules, small drug-like molecules, that could be made in theory? So the answer is 10 to the power of something, and you need to just have in your mind a guess of what that something is. Right, hope you have it. Was it that number? Again, we don't know, but it's roughly this very, very large number. The number of compounds that are possible to make is absolutely tremendous. And it's the medicinal chemist's job is to try and figure out which compound in this huge sea should I make next to try and progress my project forward and find a more optimal compound. Whereas if we compare that with the number of compounds that are actually enumerated in a database, we have a structure, maybe we've got some activity value for it. This is also a 10 to the power of something. Think about what that might be. Hope you have that guess. It's roughly this number, which is only that. So much, much smaller. And at least it's a start, but of course, there's a lot more chemistry that can be explored. And so the hope with bringing AI to drug discovery is that we can both explore more compounds overall than we might have otherwise been able to purely looking in the lab, and hopefully also shorten that time for development of finding the optimal compound sooner than the traditional amount of time. And just to highlight what we've seen over time as well is that the number of AI-inspired compounds that are entering clinical trials has really been on the rise over time. That's maybe a bit of a gray area because what does it mean to be AI-inspired? Did we ask ChatGPT something at some point, or did it really drive the program? But nevertheless, there's definitely this marked increase in the number of compounds that have had AI play a role. All right, so let's start talking now a little bit about AI itself and just some general overview and concepts. So we'll start with programming. Programming is a general field where what one does to solve a problem on a computer traditionally is you have to tell the computer very explicitly, step by step by step, add these numbers, then divide, then change them, very explicitly how to solve a problem. Within this set of programming, we find artificial intelligence, which is actually a very broad term, and it's defined as anything that simulates human logic. And so that could be a robot that's picking up something, for example. But what we're more interested in discussing today is really this branch called machine learning, which is a set of algorithms that learn patterns in your data and then can hopefully provide useful predictions going forward for unseen data. And just to emphasize why machine learning and programming is different, as I mentioned, with classical programming, what you normally have is some sort of input data that you're starting with and some rules, some processing steps that you have to explicitly define upfront in painstaking detail how to go about solving the problem you want to solve. And that will produce your output. However, with machine learning, what is very useful is that generally what you can do is you can come with a data set that has a bunch of inputs and corresponding output. So you know the goal that you want to get to, but you don't actually have to know necessarily how to solve the problem step by step. And what the algorithms do is they look at the inputs and they look at the outputs and they find the rules that help you connect those two. So for example, let's say I've got let's move away from chemistry for the moment, or drug discovery. Let's say I've got a task where I want to classify some pictures of cats versus dogs. And if I were to do this in a very classical programming approach, I would have to define some sort of rule structure that I might look at to differentiate these. So just as a dummy example, maybe I would first look at something like whiskers, which tend to be quite well defined on cats, but dogs do have whiskers too. So, even this isn't perfect. Maybe then if it doesn't, I will have to look at some other type of feature that I can understand and design, and eventually get some sort of classification of these two. However, with a machine learning approach, what you can do is you give the algorithm a set of inputs, which are these images, and a set of labels. So these would be the cat pictures, these would be the dog pictures. And I let the algorithm decide for itself what features of the image is most useful for making this classification. And it learns those rules, the most effective set of rules for itself, how to connect this picture with this label and this picture with this label. And so what that means is, and why it's useful is, in the traditional sense, I need to typically have quite a lot of domain knowledge and understanding of my problem such that I can craft these rules myself. And it's quite a rigid and defined approach. If there's maybe new data sets, let's say in this example, new breeds of animals, the existing architecture won't easily adapt. I'll have to write a whole set of new rules to account for that. Whereas with a machine learning type approach, I don't need to have a huge amount of domain knowledge. I don't need to know explicitly how, say, each aspect is working. Of course, if you do know some domain knowledge, that can be very helpful still in how you go about crafting your model. And it tends to be a little bit more abstract, and you can cater for more complexity in your data set because the algorithm is deciding for itself what is the best way to connect the input and the output. And as I give it more and more data, this tends to help the model learn better the patterns. So if we shift for the moment to drug discovery, back to drug discovery, you can do the sort of analogous breakdown where perhaps if I have my initial set of molecules and I'm trying to predict if they're going to be active or inactive, I would first have to describe them somehow with a very human interpretable understanding of them, and then perhaps use some logic-based rules that I can understand. So maybe I would say Lipinski's rules of drug likeness is maybe a good way to start breaking this down. But that's not very easily adaptable, especially if the biology, say, is very complex. And so with machine learning, what I can instead do is bring my molecules, bring their labels, active, inactive, and ask the machine learning algorithm to find the patterns between the structures and the activity, so that when I come with new structures, it can make a prediction. And I don't necessarily need to have deep understanding, technical understanding of how these actual molecules, say, are binding to their proteins and how they're being selective. As long as I can give the model enough examples, it can then figure out those patterns. All right. So now we're going to start moving a little bit more deeper and deeper into the AI side. And really what we're going to focus on initially are considerations around data. And this will be the activity that we do a bit later as well. And so I can't emphasize enough how important the quality of your data is to the model building process. You could have the best, most sophisticated model training framework ever. But if you put in data that's very messy and noisy, it's not going to be able to make sense of that, and you're going to get nonsense results. So this garbage in, garbage out paradigm is something to keep remembering. We're trying to make this data as clean as possible so that we can let the model identify and learn the patterns as efficiently as possible. And so the rest of this talk will be framed around these general steps of what it takes to train a machine learning model. Again, this is not for you to be able to train your own model. We're not going to that technical detail, but it's to understand what's involved in this process so that when you read papers or you interact with data scientists, you can understand what is going on, and more importantly, how do you actually use the models going forward. So as data scientists, we spend a lot of time in these initial two steps, this data curation, so finding the data, and then cleaning the data so that it's not garbage in, garbage out, but there's actually something to learn. Probably 70 to 80% of one's time is just spent on these steps, really processing the data. And then the fun bit is only, say, 20% of the time, maybe. So where do we get this data? Of course, if you have your own data, that's fantastic. It will be really relevant. If you need a starting point where maybe you don't have all of the data and you need something a bit more general, then there are numerous databases out there, especially for chemistry and bioactivity type data. So a good first port of call is the Chembl database, and we'll see a lot of that in this workshop. But there are definitely others. So COAD, for example, does a lot of work for the escape pathogens. Therapeutic Data Commons have some data sets that are also used as a benchmarking set. And so these are good sources to find your initial set of data. Once you have your data, now you want to start going through the cleaning process. And what do we mean by cleaning? Well, in general, we're talking about concepts around these 4 points. There are some others, but these are the key 4, where we want our data set to be complete. So we don't want any missing values. We want our data to be accurate. So if there's noise in the data, that means it's harder for the model to learn what it needs to learn. And so you're going to get a less predictive model. So we're always thinking here about signal and noise. How much signal is there in the data and how do I maximize that signal? So here you can imagine if I have an assay, but the readout has a 10-fold variation between measurements, that's a very noisy readout, and your model is going to struggle to differentiate then between your different categories or how to predict those compounds. Compared to, say, an assay, maybe if it's got a 2-fold variation, there's a lot more focused signal for the model to learn from. Ideally, we want our data to be as consistent as possible. So we'll talk about some examples, say, what if you're measuring activity of a molecule, but in 2 different media conditions for assays? How is that introducing noise into your data sets? Is it something to maybe split out and isolate each individual assay to really maximize the signal coming from that? And then also, all the data needs to be relevant. So it needs to actually pertain to the problem you're trying to solve. And in general, what we're looking for is we want a unique set of inputs. And if they have labels, they each correspond 1-to-1 to an output label. So we don't want multiple inputs with duplicate compounds with the same value, and we don't want one compound that's got multiple labels. We want a 1-to-1 relationship. A few more points to mention. Of course, the more data you have, the more opportunity you're giving the model to learn the patterns. If you have very few data points, it's going to be very difficult for your model to generalize across more chemical space. The natural question is always how many compounds do you need to train a model? And that's quite difficult to answer, but normally a good starting point and minimum, absolute minimum would be, say, 30 to 40 compounds, but ideally you would have hundreds if possible. And also the characteristics of that data set is you should have examples of both good and bad molecules. If you only gave it all inactive molecules and you said, tell me what an active one will be, well, it's not magic. The model is just using maths to find the patterns, and you haven't given it any examples of what to actually look for. And so always be thinking about what have I given my model to learn, and what does that mean that it can be used for? And then last thing to just keep in mind is as you are training models on different types of data sets, is just to think about how much complexity is in the assay. So for example, if I've got a protein inhibition assay, that tends to be quite a standard and clean assay. There's just the protein in a dish, and we're just checking if the molecules go in and bind. Compared to a whole cell activity assay, there are much more factors going into that. Is it actually permeable through the membranes and getting where it needs to go, for example? And that doesn't mean we can't model the data, but it just means that there might be more noise in the data than say other data sets. So as I mentioned, what we're really looking for is if we go from this raw data set that I've pulled from Kimball, for example, which actually is pretty good with a lot of the processing steps that they do. What I'm looking for to be able to build a model is have a data set where I've got a set of input structures and a set of values. And I'm going to ask the model to learn the patterns that correlate structure to value. So now I'd like you to, again, just think about your own experience and your own data. Think about the readouts and the data sets that you get from your different experiments and try and identify some issues that you might need to go and look for if you were thinking about using that data set to train a model. And this will be really important. This will be part of your activity a bit later. So I'd really encourage you to think about this, right? So some that I can think of off the top of my head is, well, what if you've got multiple measurements for the same compound? And what if those measurements disagree with each other? Then what do you do? Do you take one result? Do you discard the compound because it's uncertain? You need to be aware of missing values, where maybe you only have a structure, but you don't have the label if the rest of them do. This is a common problem where assays will only measure, say, activity or solubility in a certain range of measurements. And very often it happens that a compound's real measurement falls outside of that range. So you might get a value that's, say, bigger than 20. You don't know if it's 200 or 2000, you just know it's bigger than 20. And so is that an issue? Sometimes you need to take that into account. We'll talk a lot about this as well. What about if your compounds have been measured in different assay conditions or different readouts were used? You need to be able to standardize that. Otherwise, again, you're just increasing the noise in your data sets and not giving enough signal for the model to learn from. Our compounds in text are represented by this structure called smiles. And smiles, you get multiple smile strings for the same compound, just depending on where you start labeling the atoms from. And so there are ways to standardize this, but if you don't standardize it, then you might actually have duplicate values and you just weren't able to detect them. And then, of course, considering care if you've got many outliers, different chemical space, or very active versus or very inactive, how do you deal with that? Is it a problem for your model? So these are all examples that you would maybe want to look for that would affect whether the data set is complete, accurate, consistent, and relevant. All right. So now we're going to move on a bit. We've got our data set now. We've cleaned our data set, and now we want to start applying some techniques and methods. Well, the first thing we need to do is this process called molecular descriptors or featurization, where you and I, as humans, we can look at the structure and we can very easily interpret what it means, but a computer cannot. A computer works on numbers. And so we need a way to convert our chemical structures, which are represented as smile strings, into some sort of numerical representation. And so here I've given the most simple example. For example, if you have a bit vector, which is a vector of zeros and ones, and every position corresponds to the presence or absence of a particular chemical group. And then if I take my molecule and I say, okay, does it have a phenol ring? Well, the third position corresponds to phenol ring, so I'll put a 1 there. Or does it have a hydroxyl group? Okay, if it does, I'll put a 1 in the sixth position. And so this is a way to represent your molecule using numbers. And there are many, many different ways of doing this. So this is just the most basic of examples. And you'll often be thinking about, well, what type of information has my featurization approach captured? So there are many different ways, perhaps starting potentially even more simple than before. You could capture some sort of global properties, so molecular weight or just number of carbon atoms, for example. Moving up in complexity, perhaps you start looking at local substructures or calculate some sort of matrix of all-to-all atoms where you're calculating the electrostatic force from every atom to every other atom. Maybe that gives you a little bit more information about the three-dimensional characteristics. And then this is perhaps where you start moving up in complexity, trying to get more, say, Cartesian coordinates or define three-dimensional characteristics of your molecule. You can include maybe time domain information, how your molecule might have different conformations or interactions with its targets. Is this an interaction with its target to be aware of? And then in more recent years, we've had these so-called data-driven descriptors where you train these huge neural networks, even beyond just chemical data, clinical data, biology data, and allow it to learn a representation that is useful for itself. As you tend to go up these orders of complexity, you're going up in computational processing costs. And also, as you tend to go up in complexity, it becomes less and less interpretable to a human, potentially more useful for the computer algorithm. And so we tend to balance these, how complex we go. But I will describe one which you'll often see in a moment in different papers. Just before we get there, something else you might have heard or will see in papers is this concept of unsupervised versus supervised learning. And all this means is, does your data have labels? And for a chemistry data set, the chemistry compound structures are the minimum that we need. And if we don't have any activity data, we can still do some analysis around that, and that's what we'll talk about next. And that's what we call unsupervised learning, which means the data set has not had labels by a supervisor. Compared to supervised learning, where you actually do have labels. So this could be solubility, it could be activity, 1-0 for active, not active, or an actual IC50 value, how potent it is. And here, so this we call supervised learning, where the data has labels. So in either case, we still need to describe our molecules. We're going to talk about the unsupervised case now, and something you'll see very often in literature for models is these things called Morgan fingerprints or extended connectivity fingerprints, ECFP. And all that is at a very high level, not to go into too much detail, is to say that what is happening is it looks at each atom in your molecule and it looks at their immediate neighbors up to a certain radius. And so each of these correspond to a particular chemical substructure. And if you do this for all atoms up to the radius size, and you count how many of these substructures and put them into a vector, you've got a Morgan fingerprint. So each of these positions in the vector will correspond to a particular substructure and the number of times it appears in the molecule. And typically this vector will be 2048 dimensions, or 2048 bits. So now if we start thinking about how do we actually use this representation of our molecules, if we have an unsupervised task, so we don't have labels, well, the common thing to do is to plot this data to visualize it. But as humans, we really struggle to visualize anything above 3 dimensions, right? So 2048 dimensions of data, we can't very easily visualize that. And so we use a set of techniques called dimensionality reduction, where we want to go from our high dimensional space down to just a few dimensions where we can plot our compounds and see how they relate to each other. So one way that you might see in many papers to do this is something called PCA, which is principal component analysis. And we'll go through this in a different example in a second. And don't worry if you don't understand the details, but I'm going to describe it here for interest's sake, which is what happens is you take your data set. Here's just already 2 dimensions, but to show you how it works, we look at our data set and we find a new axis through our data set, which maximizes variance. And then if you want a second axis, you look for something perpendicular that maximizes the next amount of variance. And you can keep doing this to as many dimensions as you need. Here, we're interested in doing 2 dimensions because we want to plot this data. So let's go up 1 dimension in our raw data. If we have 3-dimensional data and we want to apply a PCA to this, what we do is we find our primary axis that maximizes the amount of variance around that axis. And then I can find a second axis perpendicular that maximizes the next amount of variance. And therefore, in this case, I'm going to stop at just 2 axes. And what I can do is project my raw data in 3 dimensions down into the new axis set, the new principal components axes. And this allows us to very easily understand or look and interpret our data, see if it makes sense. And so just to re-emphasize, what we've done is we've gone from our featurized description of our molecule, which consists of many numbers, and we've gone down to just a few numbers, which still describe this molecule on these new axes and help us to understand how it relates to other compounds. So let's go to a more modern technique. So this is a technique called UMAP, Uniform Manifold Approximation and Projection. Something similar you might have heard of as a TSNE plot, but both of these are non-linear transformations. In our PCA, these are linear, so we're just rescaling the data down to our new axes and maintaining all of the relationships that they had in the high dimensional space. So a point that's twice as far away will still be twice as far away in the PCA plot. Whereas with the UMAP, what we do is in our high dimensional space, we link together the compounds that are most similar to each other. And once we've projected them down into a lower dimensional space, we redistribute those points to maintain those connections in this new space. And so we're changing the underlying relationship of the global data in order to maximize the visualization aspect. So really, you can think of this a bit like a clustering type approach, where it's emphasizing these local relationships rather than the global structure of the data. So here, for example, is the same data set that's been transformed through PCA and UMAP. There's not necessarily one way that's better than the other. Often you would look at both, for example, and that might tell you different things. And just to emphasize again, so the PCA really maintains the global structure of your data. You've just linearly scaled down the data to new axes. But perhaps if your data is very complex, it's kind of difficult to understand. So for example, if we think about these dark orange versus the blue dots, are they the same? Are they very different? How do they relate? Certainly the dark blue dots are very different to everything else here. Whereas in the UMAP, we've changed that global relationship to maximize the interpretability. And we've clustered these compounds so you can see, okay, the blue dots and the orange dots, well, it actually thinks these light blue dots are more similar to these guys, and the dark orange less so. And the fact that these are close together, they're still maintaining some similarity. And so overall, what you're looking for in these types of plots is the closer they are together, the more structurally similar those compounds are, versus if they're far apart, the more dissimilar they are. All right, so let's go through some examples to unpack that to make it clear. One such data set that we can look at, if you're not aware of, is something called Coconut. And this is specifically a natural products database. So for this next example, what I've done is pulled some data from Chembl, drug-like molecules, and compared it to some data from Coconut, which are predominantly natural products. And so what we see in our plots, you can see Chembl in red and the natural products in blue. And I think what we see makes sense. What we see is that the drug-like molecules are all very similar to each other compared to how different the natural products are, which is expected because natural products tend to be more chemically complex. And what we see in the UMAP is that we seem to have some local clusters of data. So perhaps this represents analogs around a similar scaffold. but these guys are different to the central mass of compounds, and certainly less similar to the Kimball compounds because they're further away. So now I would like to challenge you and get you to think a little bit. So let's say I've got a data set. I'll call them the black compounds and the red compounds. The red compounds are all compounds from one chemical series, which means they share a scaffold and they just have variations on the outside of the molecule. And the first question is, if I trained a model only on the red data points, how well do you think I would be able to predict the black compounds? So hopefully that's not too complicated. Your model is very focused in this local region of chemical space. And so to expect it to be able to generalize to this much wider region is probably not realistic, and you're probably not going to get very accurate predictions. And let's flip the paradigm. So let's say I train a model on all of the black compounds, and now I want to think about how well is it going to do at predicting the red compounds. I'll give you 5 seconds to think of an answer. So this one's a little bit more tricky, I would say. Certainly it's a better case than the first way around. The black compounds are quite diverse, and the red compounds are within this chemical space. But these are very, very fine chemical changes, and these are much faster. And so perhaps it will struggle a little bit to get that really fine-grained chemical information. And so all of this is getting at this concept called the applicability domain of models, which is thinking about what has my model seen when it was trained, and how do I want to use it for data that I'm interested in? Are those similar? And it's not to say that it will be completely irrelevant if they're dissimilar training spaces, but the main concept is that the more similar your training data is to your testing data, the more confident you can be in your predictions. And so to summarize the unsupervised learning side, the types of things you can ask with this is you can explore the structure of your data set. So how clustered is it versus how diverse and homogeneous is it? Do you have outliers which you maybe need to be aware of? You don't have any labels for this data. You're just purely doing this on structure, but it can be very informative. And particularly, a lot of, say, what I would use it for is checking, is the training data relevant to the test data that I'm interested in? All right. So now moving into the last section, let's just remind ourselves we've got unsupervised learning, which we've just spoken about, PCA, UMAP. This is where you don't have labels on your data, so you don't have solubility measurements or IC50 values, you just have structures. And now we're going to talk about the supervised approach. So here we've got both structure and label. And this label could be a binary score, so a 1 for an active and a 0 for an inactive, or it could be the actual experimental measure, and that will be relevant shortly. So what we do is the general paradigm is we have our training data set where we've got chemical structures and we've got associated labels. So let's say the red means it's active and the blue means it's inactive. So that will be a 1 or a 0. And the first thing I always need to do is featureize my molecules. So convert them into a number representation that a computer can understand. And I feed it to a machine learning algorithm that learns to correlate this numerical description with the corresponding label. And once I have my model, what I then do is, what I'm going to use it for is to come with a compound where I don't know the label for it. So I've got a design idea in my brain, and I want to know, based on what the model has learned, does it think that it's going to likely be active or inactive? So I have to follow the same featureization approach and get a numerical description of this query compound. And then I ask my model to make a prediction. Do you think, in this case, it's more likely to be active or inactive? What we actually do during our model development process is we take our training data and we split it into different sets. So we've got our training compounds, which is what the model is really going to learn from. And then I hold out some data which the model doesn't get to learn from, but I'm going to check the model to see how well it's actually done at learning. So if you can imagine, if you're writing an exam paper and you've done some past papers and studied really hard, and then you get into your exam venue and you sit down and you look at your table at your exam script, and the questions are exactly the same as the ones from the past paper. Well, in that case, the university can't really assess if you've actually learned something or if you just have a really good memory. So here what we're doing is we're holding out data that are similar to our training compounds, but not the same. We let our model learn from the training data, and then we ask it to predict these compounds, which we know the true values for, and see how well it did at learning them. So we get a real estimate of confidence. How much has the model learned? How well has it been able to generalize? We then typically do this training check quite a few times as we're investigating maybe different ways of featurizing the molecules or different modeling algorithms. And then what we finally do is we hold out this test set, which is the final set of compounds to say my selected model, my final one that I'm happy with, that seems to be working well, does it really work on another fold of data that I've held out? It didn't just happen to do really well on this split of the data. And particularly when you've got low data scenarios, which is often the case in drug discovery, this is where you spend a lot of time in your modeling efforts to make sure that your model has really generalized and not just gotten lucky with the splits of data that you've done. Once you've done this investigation where you've trained and checked and trained and checked, and then finally done your final test, and you're happy that the model really seems to be learning something general consistently, then you'll take your full training set as your final training set for your model. You can't test this model as thoroughly because you haven't got external or separate data, but now you want to give your model the best chance at learning as much as possible because you're confident that the way you're approaching it is working well. And you'll have your final model, and this is the one that, for example, I would give to the chemists at H3D to make predictions for the molecules. They'll actually go and make those molecules in the lab, and then I can go and check, hey, did all of the predictions, did they line up with reality? To what extent? Very important to note, no model is going to be perfect. You can't have that expectation, but if it's providing a good prediction, it's quite accurate, then there is a lot of value there. It's helping you enrich how many hits you're making and decreasing the number of inactives you're making. All right, so we've finished the model training journey. I'm going to finish with just a couple of slides to show you. We've spoken about unsupervised learning here. We're going to complete the picture with our supervised learning. And this is really what you're going to focus on a lot tomorrow. But in the supervised learning, we've got labels, and those labels can either be binary category, active, inactive, or some sort of continuous value. And that determines what kind of model we want to train on it. Of course, if you've got the continuous values, you can binarize them into the categories. And so classification models are always relevant. Here, what the model is trying to do is, if this is our chemical space, it's trying to learn how to separate chemical space so that you split out the circles from the crosses, let's say the actives from the inactives. Versus a regression model where you're actually trying to learn the exact continuous label. Is it 20 micromolar or 30 micromolar or 30.1? You're actually learning that, trying to predict that actual label, the continuous value. This tends to be more challenging. And you tend to need a bit more data to do it, but you'll often see variants of both of these approaches in literature. So it's important to be aware of them and how they work, so that when you start wanting to use a model, you understand what they've learned and what kind of things they're predicting. So final couple slides, if we do a classification model, going back to our training paradigm, we've got compounds, structures, and their labels are, in this case, categorical values. So 1 for active, 0 for inactive. I do my featurization, and what the model is trying to learn is, how do I correlate this numerical description with the categorical class 1 for active, 0 for inactive. And we'll talk about what kind of output this model gives tomorrow. However, compare this to a regression model. Same process. I've got my molecules. They've got a label, which is now a continuous value. I featureize my molecules as usual, and I ask my machine learning algorithm to learn how to go from this numerical representation to an actual continuous value. So is it 0.2 micromolar or 3.4 micromolar, etc. And here we often actually work in PIC50 values with the log transformation, which tends to be more informative for a model. And again, we'll talk about this tomorrow. So then just to start wrapping up, what we've spoken about in the supervised learning side is here you've got label data, and what you're asking the model to predict is in the classification model, how likely is my compound going to be active? So let's say I've got a protein target and I've defined a cutoff of 100 nanomolar, and I want to say, here's a new structure. Do you think this is likely to be in the less than 100 nanomolar category or more? Or you could predict toxicity, solubility, anything where you've got this label. So it's quite a general approach. If you've got a regression model, then you're actually trying to predict, say, for solubility, is it 50 micromolar solubility or 100 micromolar, 102 micromolar? And ultimately, what these should be guiding you towards is you can throw many ideas at these models and prioritize which compounds you're going to go and work on next in the lab, so that you're getting an enriched set, you're finding more actives more quickly, or if you want non-toxic compounds, you're finding the inactive compounds more quickly. All right, so that's a wrap for the day one content. Let's just recap on what we've talked about. The slides will be available. I encourage you to maybe go and read them or listen to this lecture again, just to make sure you really have all of the concepts grounded. We spoke about why AI can be useful for drug discovery and why drug discovery is so hard. We spoke about what AI actually is and how it works, and hopefully that's demystified it from this magical concept that we always talk about AI this and AI that, to something more mathematical. We spoke about cleaning data, and really it's difficult to emphasize enough how important this is. Having clean data is essential. And so me as a data scientist, I'll very often go and speak to our biologists, make sure I understand the data that they're producing, and go speak to the chemist, understand how they want to use the models, so that we're really processing the data in a way that makes sense. Then we spoke about chemical space and how to plot this, and this was an unsupervised learning approach where we don't have labels and we can see the structure of our data sets, how homogeneous or heterogeneous is it. And then we finished looking at the full model training overview, finishing with supervised learning, where we actually train these classification and regression models, which is really going to be the focus of tomorrow. So I've put a few resources here. Acilio do a lot of training, especially for African scientists or the global South. And so I encourage you, if you are interested in perhaps even getting more technical, learning some coding, there are some links to different resources you can go and look at. And now what we're going to do is we're going to move into our little exercise. So I will leave the slide up on the screen. Let's see how we're doing for time. Yep. So this is just going to be a data cleaning exercise to get you used to looking at chemical data and identifying potential problems that you need to look for. So Nicola has sent you an email with this data set in it. The scenario that we're painting is that we are interested in predicting activity for a pathogen called Acinetobacter baumannii, which is a very pesky bacterial or bacteria. So we have this data set from this database, and they have a number of compounds that have been tested for activity against A. baumannii. And before we can even think about plotting this data or building models, our first step is to go and check what kinds of things do we need to look at for this data to be able to clean it. And so for the next 15 minutes, I would like you to go through this data set and identify as many aspects of incompleteness or inconsistency or inaccuracy that you can find, and put it in a Word document. And the link to send it to, if you go to this link where all of our resources are, there'll be a link to submit your day one exercise. And so I'm going to give you until 10 past, maybe 12 past 1 to do this exercise and submit your answers. And just a hint, one very quick and easy way to do this if you don't know how to code, which is fine, is you can often find these types of inconsistencies if you sort your columns. So for example, maybe I sort by this value or by the units, and you can maybe then see which, if you've got multiple or not. So that'll be a quick way to identify some problems in this data set. There are quite a few. So with that, I'm going to mute my microphone and my camera and let you get on with this until 10 past 1, and then I will come back on and we'll discuss the exercise. All right, everyone. So it's time to start finishing up. I'll give you 2 more minutes. So maybe make your way over to the link and submit your form. It's really fine if there's just one answer or 2 answers. Just want to see that you've engaged. So I will be back in 2 minutes and then we'll talk through the exercise. And if you can't submit it right now, then please do so before the class tomorrow. Thanks. Great, everyone. So a lot of content today. Let's start wrapping up this exercise. I'm going to close this slide and we'll talk through what I found, for example. So hopefully you guys managed to find 1 or 2 things that were wrong. Maybe you did realize that it's actually quite a messy data set and quite typical of our real world setting, where we have to do a lot of this kind of investigation to find problems in the data and resolve them before we can even think about machine learning. So let me share my other set of slides. All right. So some examples of what I found when I was looking through. So this was our data set here. Let me just bring this across quickly so we can reference it here too. Okay. So when I was looking through, well, the first thing I noticed was some of these columns weren't actually that useful for anything that we were going to use them for. So they weren't very relevant. So this index tag doesn't really have much meaning if they already have an ID associated. It had this compound name, which was for approved drugs with market names, but that doesn't actually tell us any detail about them that we can use for modeling. So perhaps you would remove that column. Here, if you remember, we were particularly interested in modeling Acinetobacter baumannii activity. But this data set has activity values against multiple species, not just Acinetobacter. So particularly, say, for the Homo sapiens, so human measurements. Obviously here, this is something we don't want activity for, because activity in human means tox, essentially, if we're thinking about infectious diseases. So here, this is not very helpful in helping us predict Acinetobacter baumannii potentially. You could maybe think a bit more carefully about these other pathogens if you're looking for a compound that is broadly active against many species. Maybe there's some value in having this data, but it's not consistent and clean, right? So ideally, what we would want if we want to model Acinetobacter baumannii is we just take the compounds that have measured activity against that species, and ignore the rest, or we build separate models from the rest. But for now, we're just really focusing on Acinetobacter baumannii. There were some compounds which had multiple entries, and this is before we've even standardized our SMILE strings to make sure that they've all been captured in the same way. We haven't just labeled the compound from different sides of the molecule, but already there were duplicate entries. Within one entry, so one SMILE structure, there was this number of assays column. And if you sorted this, for example, you would have seen that some of the compounds had one measurement, one assay value, and that was it. And other compounds had been measured up to 6 times, and the resulting value here was a mean of those. So this should raise some questions from the data scientist. Even if you can't do something directly about it, at the very least to be aware of how much signal or how much noise might be in your data, if you know about your assays, how much variance there is. So is the compounds that have only been measured once, are they reliable? For the compounds that have been measured multiple times, what happened if there was variance in the data, if there was an outlier? Is the mean capturing that outlier as well? So we've kind of skewed our data. Or if we've got the raw measurements for all 6, can we maybe capture the median or identify the outlier and exclude it and then only take the mean from that? So this was a source of potential noise. There were compounds that were measured once and others multiple times. Of course, when we say Acinetobacter baumannii, we don't just necessarily mean measuring one thing. There are different strains, clinical strains, wild type, circulating lab strains. And in this data set, within Acinetobacter baumannii, so the organism, the strain that was used was sometimes different. And so that means we might need to go have a conversation with our biologist to understand the assay conditions. For the most clean and consistent data set, we would just keep one of these. Say this is a very common strain. But if we're in a very data poor scenario, then perhaps we want to try and understand how similar is this strain? How similar is the assay conditions? Are they compatible? Or perhaps maybe this is an extremely drug-resistant strain, and in which case it's not comparable. You can't say that they're the same. If they're active in one, they're going to be active in the other, for example. And so along with similar thinking, within the Acinetobacter baumannii, there were these different assay IDs. And so there's a huge question mark here. Is this assay ID with GN034 the same as this assay, which is GN034S20? What does S20 mean? I don't know. So this is again where if you're in the public domain, you need to think about this. Do I need to exclude it to be safe, or do I have a biologist I can go speak to understand what this means and if it's relevant? A big one, if I sorted by the units in my data set, many compounds were measured in micromolar, but there were some compounds which were measured in an alternate readout, which was micrograms per mole. And these are quite different. This is roughly twice as big as micromolar if you do the conversion. And so you can think about if you are defining a classification threshold, then depending on the unit you've used, that compound might fall one side of the cutoff when it really should be on the other side. Or if you're training a regression model and your units are all different, you've really introduced a lot of noise into your data set, and it's going to be difficult for your model to really identify the patterns. And the last one that I found in this list was the limits of detection. So many of these readouts had this greater than a number. So the assay was only measured, in this case, up to 10, say, but the real activity of the compound, it was quite inactive, so it was above that value, but it could have been 11. So if what I'm defining as an active or inactive is, say, at a 20 micromolar cutoff, that's suddenly really important. Whereas the compounds that are above 32, if what I consider experimental active is less than 20, then that's fine, because no matter what it is above 32, it's always going to be inactive. So again, here you need to think about, what does this mean? If I'm really not sure, this compound could be 11 or 12 or 13, introducing a lot of noise, do I need to remove those compounds because I can't be sure what they actually are? And particularly in the case of, say, a binary classifier. So if you've got these values for a regression model where you need to train on the exact value, well, this is not possible here because you don't actually know what the exact value is. But you can think about, how do I include this in a classifier where you have these categories? So even if it's above 32 and it's a lot above 32, maybe it's just inactive as the label. So those were maybe some of the things you found. If you found other problems in the data set, then great. So there were a lot to find. And remember, what we're ultimately really looking for is to get down to that set of one input structure to one label, and that label is hopefully very accurate, never missing, etc. So please do submit your exercises still if they're outstanding. Try and find anything maybe that was not on this list, or something you thought to look for that could be a problem because of your experience with running assays, but you didn't actually see that it was relevant in this case. That's also very interesting. And so with the last few minutes of our session, we will open it up to a question and answer session. So or maybe I'll hand over to Nicola to manage that.